Hello everyone, welcome to the channel Microbial World. Today we will be discussing the topic bacterial cell structure. So before we discuss about the structure of a bacterial cell, we need to know what are bacteria. Bacteria are microscopic, unicellular, prokaryotic organisms. We say that bacteria are microscopic because they are too small that they can only be visible through a microscope. They are unicellular organisms, meaning they are single-celled organisms. And we also say bacteria are prokaryotic. It is because bacteria do not have any membrane-bound organelles. Membrane-bound organelles such as chloroplast, mitochondria, endoplasmic reticulum, or peroxisome, lysosome, or true vacuole is not present in bacteria. They lack true nucleus and nucleolus. This is the reason why we say bacteria are prokaryotic. Now, the living beings are mainly classified into three domains: archaea, bacteria, and eukaryota. Among them, bacteria is the largest domain. The study of bacteria is called bacteriology, which is a branch of microbiology. As you can see, this is the structure of a bacterial cell. Now, this bacterial cell is surrounded by several layers, which is collectively called the cell envelope. The innermost layer of that cell envelope is the plasma membrane that is surrounding the cytoplasm. And the bacteria also have a cell wall surrounding this plasma membrane. The outermost layer is called capsule, which is present external to the cell wall. Now, certain bacteria will also have slime layer instead of this capsule. The genetic material, it is localized in a region that is called the nucleoid. As you can see, there are no membrane-bound organelles are present inside this bacterial cell, and hence the interior is very simple. And there are ribosomes, larger masses such as inclusion bodies, plasmids are all scattered in the cytoplasm. Many bacteria also use flagellum for locomotion, that is for movement. So now let's discuss each part of the bacterial cell in detail. The structure of a bacteria can be studied as external and internal structures. So the external structures includes the cell wall and all structures outside the cell wall, which means it includes the cell wall, capsule, flagella, fimbriae or pili, sheath and prosthesis. The internal structures include the cell membrane and all the structures inside the cell membrane, such as the cell membrane, cytoplasm, ribosomes, mesosomes, inclusion bodies, and bacterial nucleus. So first we are going to discuss about the external structures. Under this comes the capsule. So the capsule is a viscous outermost layer surrounding the cell wall. Viscous means it will be having a thick, sticky consistency. It is having a thick, sticky consistency. Now this capsule is mainly composed of polysaccharide or polypeptides that constitute 2% and water, which constitutes 98%. This capsule, we cannot see this capsule in all kind of bacteria. Capsule is only seen in some species of bacteria. So an example of a capsulated bacterium is Streptococcus. And there are mainly two types of capsule. First is macrocapsule. The capsule which is having a thickness of 0.2 μm or more is called macrocapsule. And microcapsule is the capsule with thickness of 0.2 μm or less. So in some species of bacteria, instead of this viscous covering, an amorphous or paracrystalline protein material will be present. It is called the slime layer. Now the slime layer is a loosely organized structure, whereas this capsule, it is a sharply defined structure. Now some of the functions of this capsule is adherence. Adherence means the bacterium will have the ability to attach to the host cells, attachment to the host cells. They prevent desiccation, that is a state of extreme dryness. They resist phagocytosis by white blood cells. Now what is this phagocytosis? It is the ability of certain living cells called phagocytes to ingest or engulf other living cells or particles. You can see this phagocyte is trying to ingest or engulf another living cell or particle. Now, one of the function of the slime layer is that it provides protection to the bacterium from proteolytic enzymes. The capsule, it can be identified using capsular staining methods. Now the staining procedures, it will be discussed in detail in subsequent sessions. This capsule, it is antigenic, and hence the capsule, it can be demonstrated using various serological methods. Now, when a suspension of a capsulated bacterium is mixed with its specific anticapsular serum, the capsule will become swollen. The capsule gets swollen, and this is called Quillen reaction. Now this Quillen reaction was described by Neufeld in 1902. Cell wall. So the cell wall is what that provides shape to the bacterial cell. And the cell wall, it also makes the cell rigid. That means the cell, it does not change its shape. The cell wall, it cannot be seen by light microscopy, and it also do not stain with simple stains. Now gram staining is what? Gram staining is the procedure that is used to differentiate bacteria based on its cell wall constituents. We can demonstrate a cell wall only by electron microscopy or mechanical rupture of that cell. The cell wall, it is made up of peptidoglycan. This peptidoglycan, it is a cross-linked polymer of alternately repeating NAM and NAG. That is, it is a cross-linked polymer of alternately repeating N-acetyl muramic acid and N-acetyl glucosamine. Now this peptidoglycan, it will surround the plasma membrane as an external coat, and it is 10 to 25 μm in thickness. So based on composition, the bacterial cell wall is classified into two types, that is gram-positive cell wall and gram-negative cell wall. So let's see what is gram-positive cell wall. Gram-positive cell wall is a thick cell wall, and it contains large amount of peptidoglycan. It constitutes about 40 to 90% of the cell. It also contains acidic sugars such as teichoic acid and teichuronic acid. And it also contains neutral sugars such as mannose, arabinose, rhamnose and glucosamine as matrix substances. So this teichoic acid, it is made up of polyribitol phosphate or polyglycerol phosphate. And this teichoic acid is the major surface antigen of gram-positive bacteria. Now this teichoic acid is of two types, that is wall teichoic acid and lipoteichoic acid. This wall teichoic acid, it is linked to the peptidoglycan, whereas lipoteichoic acid is linked to the membrane glycolipids. Then there is also acidic sugar such as teichuronic acid that is present. Teichuronic acid, it is a polymer of N-acetyl mannuronic acid and D-glucuronic acid. During this gram staining, there are mainly two dyes are involved, that is crystal violet and safranin. So these gram-positive bacteria, they take up the crystal violet dye and these gram-positive bacteria appear in purple in color. This is the structure of a gram-positive bacterial cell wall. So as we said earlier, the gram-positive bacterial cell wall is having a very thick cell wall and large amount of peptidoglycan is present. So we said that this peptidoglycan, it is a cross-linked polymer of alternately repeating N-acetyl muranic acid and N-acetyl glucosamine polysaccharide subunits. So you can see here, this is the wall teichoic acid that is linked to the peptidoglycan layer, and this is the lipoteichoic acid that is linked to the membrane glycolipids. And this is the periplastic space, the region between the peptidoglycan and the plasma membrane. And this is the plasma membrane, this is the membrane proteins and the cytosol region. Gram-negative cell wall. Now this gram-negative cell wall is thin and it contains less amount of peptidoglycan. Okay, it is having less amount of peptidoglycan, and in addition to this peptidoglycan, they are having lipoprotein, lipopolysaccharide and an outer membrane. Now this lipoprotein, it is mainly composed of bronze lipoprotein. This bronze lipoprotein, it is an outer membrane protein and its function is to stabilize the outer membrane. The lipopolysaccharide is a complex molecule that is consisting of three components, that is lipid A, core oligosaccharide and O polysaccharide. Now this lipid A contains phosphorylated glucosamine disaccharides, hydroxy myristic acid and fatty acids are present in this lipid A. Core oligosaccharide mainly contains two sugars, ketodeoxyoctanoic acid and an heptose sugar that is bounded by lipid A. The O polysaccharide will have wide variety of sugars and it will differ between the bacterial strains. Then comes the outer membrane. So this outer membrane, it is a bilayered structure. It will be having an inner layer resembling the plasma membrane and an outer layer that is made of lipopolysaccharide. The outer membrane also contains sporin proteins. Now what is the function of the sporin proteins? The sporin proteins are involved in the exchange of nutrients over the outer membrane and they are also involved in pathogenesis. Now during the gram staining procedure, these gram-negative cell walls, they will lose this crystal violet dye and they take up safranin during counter staining, and hence the gram-negative bacteria will appear pink in color. So this is the structure of gram-negative bacterial cell wall. We have said that the gram-negative bacterial cell wall, it is a very thin cell wall. Now you can see here the amount of peptidoglycan in gram-negative bacterial cell wall is less when compared to that of gram-positive bacterial cell wall. This is the outer membrane region in gram-negative bacterial cell wall. This is the porin protein that is for exchange of nutrients, and it is also having involvement in pathogenesis, as we said. This is the lipopolysaccharide. Now you can see this is lipid A. This is lipid A. We have said this lipopolysaccharide. It is a very main component of the gram-negative bacterial cell wall that is made of three components, that is lipid A. Then there is core oligosaccharide and O polysaccharide. Now this is a magnified image. This is lipid A and then there is core oligosaccharide and O polysaccharide. Then this is the periplasm region, inner membrane and cytosol region. Flagella. Flagella are long hair-like filamentous structure that is 4 to 5 μm long and 0.01 to 0.03 μm in diameter. So what this flagella does is that it provides motility to the bacteria. The flagella is having mainly three parts: filament, hook and basal body. Now this filament, It is a thread-like part that is extending from the cell wall. It is mainly composed of the protein flagellum. And this hook, it is a short curved structure. It is a short curved structure like this, and this hook will join the filament to the basal body. The basal body, it is a set of rings that is embedded in the cell wall and plasma membrane. And there are two pair of rings in case of gram-negative bacteria, and there is one pair of ring in gram-positive bacteria. Some of the functions of this basal body is it synthesizes polymers of the flagellum, provide energy for evolution, and it also regulates the movement of that flagellum. Other functions of flagella is that they aid in chemotaxis. Chemotaxis is what that will help the cells to migrate to an attractant chemicals, or the cells can move away from the repellents. The flagella also helps in bacterial pathogenicity and survival. So this is a simple structure of the flagellum. As we said, the flagellum is having three main parts, that is filament, hook, and basal body or motor device. Now this filament, it is the thread-like part of the flagellum, and it is composed of the protein, that is flagellin. Then there is the hook. As you can see here, it is a curved part. It is a curved part and it is connecting the filament to the basal body. The basal body is a set of rings that is embedded in the cell wall or plasma membrane. Now, which are those rings? Let's see. There is C ring that is anchored in the cytoplasm. There is MS ring which is anchored in the cytoplasmic membrane. Then there is P ring that is anchored in the peptidoglycan layer. And finally there is L ring, that is the outer ring that is anchored in the lipopolysaccharide layer, or we can say it is anchored in the outer membrane, associated with the outer membrane. So these are the three main parts of the flagella. Pili or fimbriae. Now this pili, it is a short, hollow, non-helical filamentous structure that is 0.5 μm in length and 0.01 μm in diameter. Now, what is the difference between pili and fimbriae? Pili are hair-like fibers that is found on the surface of the bacteria, whereas fimbriae are bristle-like fibers that is found on the bacteria. Pili can be exclusively found only in gram-negative bacteria, but fimbriae is found in both gram-positive and gram-negative bacteria. Now this pili is composed of the protein pilin that is arranged non-helically. Now there is a special kind of pili that is called sex pili. The sex pili is involved in bacterial conjugation process. So what is this bacterial conjugation? It is the transfer of genetic material between two bacterial cells that occurs by direct cell-to-cell contact or by formation of a conjugation bridge. So this is a conjugation bridge is formed for the transfer of the genetic element. Now this sex pili, it is longer than the usual pili. It is 10 to 20 μm in length, and they are few in number, just 1 to 4. Sex pili is of two types, F pili and I pili. The main function of pili is adherence to the host cells. That is, it helps in attachment to the host cells. Sheath and prosthesis. Now this sheath, it is a hollow tube-like structure enclosing chain-forming bacteria. Now this sheath, it is mainly found in aquatic bacteria. Its function is that it will provide mechanical strength to the chain. Whereas prostheses, it is a semi-rigid extension of the cell wall and plasma membrane. Its function is it will increase the nutrient absorption and it also help in adhesion. So this is all about the external structures of a bacterial cell. I hope that this video was helpful for everyone. Thank you for watching.